Hey, AP Biology, it's Miss Willis, and I'm here to talk to you today about macromolecules. So you're going to learn about the four basics, which are carbohydrates, lipids, proteins, and nucleic acids. And we'll go through just the basics, the fundamentals about each, and then of course in class we'll dive deeper into each of these topics. So let's go ahead and get started. So like the name says, a macromolecule is a large molecule. There's four basic ones that you'll find in most living organisms, and those are the carbohydrates, lipids, proteins, and nucleic acids. What are they made of? Well, they're made up of a lot of atoms that are covalently connected. So that means they are sharing electrons between the atoms. A lot of these macromolecules will have the two basic elements, and that's carbon and hydrogen, and let's throw in some oxygen in there as well. Macromolecules are essentially polymers. What's a polymer? It is a long molecule that is composed of little building blocks. And these little building blocks are element-based molecules or compounds. The little blocks, we call them monomers. So remember in Latin, mono means one and poly means many. So when you put all these basic building blocks of monomers together, you get a polymer. So monomers compose polymers, and a macromolecule is a really big polymer. When you put two monomers together to make a polymer, water is lost in the process, as you can see here in this animation. So because water is lost when the two monomers bind together, we call that dehydration synthesis or a dehydration reaction. Remember, just like in running or sports, right? When you become dehydrated, you are losing water. So the same thing is happening here. When you put two monomers together to make that polymer, water is lost. And the process can be reversed. So when you want to break up a polymer, so you want to break up this long chain of monomers, guess what you do? You add water. So adding water to break up that polymer, that process is called hydrolysis. Hydro is water, and lysis in Latin usually means to cut or to split. So you can figure out the process just by breaking down the word. So remember, when you want to break apart those polymers, you add water. When you want to build the polymers, you take away water. The first macromolecule that you will be encountering in biology class are the carbohydrates. The carbohydrates are composed of carbon, hydrogen, and sugar, and they pretty much fuel the body with energy. That's their main purpose. So the two different types we'll talk about are the sugars and the starches. There are many types of carbohydrates. So we have our monosaccharides, disaccharides, and even polysaccharides. Now, monosaccharides, they start off with just the one sugar. So glucose, galactose, those are all great examples of a monosaccharide. When you put two of them together, you get a disaccharide. So a good example of that would be lactose. When you put glucose and galactose together, you make the sugar lactose. And of course, when you join those two monomers together, you have dehydration synthesis that occurs, so a water molecule is lost. What holds the two monosaccharides together to make a disaccharide? That is what we call a glycosidic bond. It's a covalent bond between the two sugars. And of course, if you put a whole bunch of disaccharides together, you will get a polysaccharide. But every time that you do that, you will lose water when you join a monosaccharide to something else. The polysaccharides are made of a whole bunch of monosaccharides put together. And two great examples that you're going to see in biology are starch and glycogen. Starch is a plant polysaccharide that is made from putting a whole bunch of glucose monosaccharides together. Glycogen you'll commonly find in animals, and we use it as an energy storage system. You can commonly find glycogen in our liver and muscle cells. And there are structural polysaccharides. So structural polysaccharides are ones that you find in the support structures of systems. So a good example of this is cellulose. Cellulose is a structural polysaccharide found in plants, but what's really interesting is humans, when we eat this cellulose, we cannot digest it. We have a symbiosis with bacteria in our intestines to help us break it down because we lack the enzymes to do so. Now on to the next macromolecule, and that would be the lipids. The lipids are also known as fats, and they are composed of also carbon, hydrogen, and oxygen, but they are shaped in a different way than when you compare them with carbohydrates. Their main function is long-term energy storage, and they tend to be hydrophobic, which means they do not like water. And the reason for this is because they have a lot of carbon and hydrogen bonds that are occurring in this molecule, and they are bonded non-polarly, if that is a word. So that means these electrons are being shared equally, and there are no tugging on each other's electrons. A lipid is usually composed of a glycerol head, so you see that here in the left, and then they have usually 3 fatty acid chains that are connected to them. And you can see, again, the main elements that are involved are the carbon and hydrogen in the glycerol, and then carbon, hydrogen, and oxygen in the fatty acid chain. When the glycerol head binds to the fatty acid chain, a dehydration synthesis reaction does occur where water is released. That bond is what we call an ester linkage. And because glycerol does bind 3 times to 3 fatty acid chains, that means 3 water molecules are released. There are 2 different types of fats or lipids that you've probably come across just in your everyday life, and that's the saturated and unsaturated fats or fatty acids. If you just take a look at the pictures there, you can kind of see the difference between the two. With a saturated fatty acid, carbon is bonded to every single hydrogen possible in that arrangement. So there are no double bonds like you see with the unsaturated fatty acid. So pretty much the carbons in the saturated fatty acids are combined to hydrogen atoms, whereas in unsaturated you may see 1 or more double bonds with the carbons in that fatty acid. So the really only difference between the two is the chemical arrangement of their carbons and hydrogens. Now, in biology class, you're going to see these a lot. Phospholipids are a unique type of lipid. They are composed of 2 fatty acid chains and 1 glycerol, but the glycerol and the fatty acid chain is connected to what we call a phosphate head, PO4. And because of that, the phosphate head is hydrophilic, so that means it will attract water. It is a polar molecule. However, the fatty acid chain remains nonpolar, and because it's nonpolar, it doesn't like water, so we call it hydrophobic. So because it has both a hydrophobic region and a hydrophilic region, we call this amphipathic. The third macromolecule that we'll talk about are the proteins, and the proteins are composed of 4 elements: carbon, hydrogen, oxygen, and nitrogen. Now, essentially, all proteins are coded for by our DNA, our genetic information, and they can be found in pretty much every single function of your body. You may have heard of enzymes. Enzymes are proteins that speed up reaction rates, and a good example of one is lactase. Lactase is a protein that helps you break down the sugar, the carbohydrate, lactose. Also, keep in mind that all proteins are usually composed of amino acids. And so what you do to get a protein to be made is you put a whole bunch of amino acids together. So remember, one amino acid, when you bind it with another amino acid, water is lost in a dehydration synthesis reaction. But then also in forming those two amino acids together and bonding them, they create what's called a peptide bond. And if you put a whole bunch of those together, you get what we call a polypeptide. And once that polypeptide does take on a certain shape, which I'll talk about in the next slide, we call it a protein. There are four basic structures to a protein. In primary structure, you just have a chain of amino acids. So they're all being held together by those peptide bonds. In the secondary structure, those bonds begin to bend, and they can make things called alpha helices or helix or beta pleated sheets. Continue the bending of that chain and you get what's called a tertiary structure. Now this bending will usually happen because the groups of, specifically the R groups of the amino acids that are in that chain, they start reacting with one another. So based upon their properties, whether they're polar or nonpolar, they will bend in certain directions. And then sometimes proteins just stop there at the tertiary structure, but some do join together with other polypeptide chains and they make what's called the quaternary structure. So hemoglobin is a protein that carries oxygen throughout your blood, but that is a good example of a quaternary structure for proteins. We are on to our fourth macromolecule, and that is the nucleic acid. Now, nucleic acid, its main function is to store and transmit genetic information from one generation to the next. This information is coded into what we call genes, and a gene is just a section of DNA that codes for a trait. And you can find these genes on chromosomes. So we do have 23 pairs of chromosomes in our cells, and the traits are passed on from generation to generation. Nucleic acids are composed of 5 main elements. So it's carbon, hydrogen, oxygen, nitrogen, and throw in some phosphorus. And there's 2 different types that you probably have already heard of. And that is RNA, ribonucleic acid, and DNA, which is deoxyribonucleic acid. There are some differences between the two. For example, ribonucleic acid is single-stranded, where deoxyribonucleic acid is double-stranded. The main difference in the chemical formula is that DNA, because it's deoxy, it's lacking 1 oxygen when you compare it to the ribonucleic acid sugar. So that's some of the main differences. We'll talk about more of the differences later. Nucleic acids are composed of monomers called nucleotides. And so it's a little bit different between the two. If you are DNA, your nucleotide has a phosphate, so PO4 group, a deoxyribose sugar, and a nitrogen base, which is usually adenine, cytosine, guanine, or thymine. However, if you are RNA, you have a phosphate head, just like DNA, but your sugar is composed of ribose, that's the carbon sugar there, and then the nitrogen base can be either nitrogen, guanine, cytosine, or uracil. So RNA does not contain the base thymine. Okay, well, I hope that was helpful, and I will see you all in class.